Welcome to today's webinar, in which I'm going to discuss the application of lipidomics in population health studies, as the title already suggests. So the first population health or cohort study, which led to conclusive insights into associations between lifestyle habits and disease and mortality risk, was the British doctor study. It started in 1951, and it was a study among British doctors and provided first convincing statistical proof that tobacco smoking increases the risk of lung cancer. The study showed that smokers die earlier than non-smokers, and that quitting to smoke is beneficial for your health. It extends basically your lifespan. In summary, the study provided conclusive evidence of a linkage between smoking and lung cancer, myocardial infarction, respiratory diseases, and other smoking-related illnesses. In total, smoking decreases lifespan up to 10 years. And the earlier you stop smoking, the better it is. It's also an important outcome of the study. Another famous cohort study is the Framingham Heart Study. It was started in 1949 and led to the Framingham Risk Score, which estimates the 10-year cardiovascular risk of an individual. The Framingham Risk Score is based on age, sex, smoking, blood pressure, and the status of lipid metabolism as assessed by measuring blood high-density lipoprotein and cholesterol values. This already highlights the importance of lipids in lipid metabolism and disease etiology. But lipids have not only received attention in the context of cardiovascular diseases, but also are an important aspect in many other disease areas. And this is actually highlighted by the numbers of publications dealing with lipidomics. So I really like to show the slide. So since 2010, we see a 15-fold increase in publications using the term lipidomics. Lipids are a structurally very heterogeneous group of molecules. They usually consist of an apolar part, shown here, which is usually made up of fatty acids, and a polar head group, this one here. For example, in glycerophospholipids, we achieve structural diversity by different fatty acids with different fatty acid chain length, like here, and different numbers of double bonds, as indicated here. Additionally, the polar head groups here give rise to different lipid classes, such as, for example, phosphatidylcholine or phosphatidylиnositol. And in this way, thousands of different lipids can be produced by our bodies. Where do we find those lipids? So as you all know, lipids are nutrients in our diet, and once they are eaten, they enter our bloodstream in structures that are called lipoprotein particles, shown here. These lipoproteins consist of an outer shell of phospholipids, here in dark blue, and a core of neutral lipids, the triglycerides and cholesterol esters usually. The lipoproteins are the vehicles that are used by our body to transport lipids to various organs, where they serve as nutrients and building blocks for cellular structures in the form of membranes. And these cellular membranes, as you all know, surround our cells and form intracellular structures like the organelles. But not only do the lipids form these boundaries, these surroundings, but they are also the medium in which the transmembrane proteins are embedded. About 30% of the entire proteome are transmembrane proteins, actually, and many of them play important roles in cell-cell interactions and signaling processes that are really fundamental to disease mechanisms in, for example, diabetes and cancer. The way lipids are approached analytically has changed quite a bit in the past years. It all started with thin layer chromatography to analyze lipid composition on the lipid class level. Then, with the advent of mass spec-based lipid analysis technologies, as depicted here, it became possible to really analyze lipidomes at the level of individual lipid molecules. At Lipotype, we have established a shotgun lipidomics technology for the quantitative analysis of lipids that basically consists of two essential parts. The first part is the lipid analysis itself, and the second part is the data analysis. So I will now briefly walk you through the whole process to give you a technical overview of how we do the things here. So it all starts, of course, with the lipid extraction, which is sample type specific and done fully automated in a 96-well format. The lipid extracts are then infused directly into a mass spectrometer by means of an robotic ion spray source, which is also working in 96-well format. The mass spectra are then acquired on a thermo-Q-exactive mass spectrometer, which features Orbitrack technology and has a very high mass resolution that really allows us to resolve very complex mass spectra. When it comes to data analysis, down here, we apply proprietary software for both lipid identification and also statistical analysis. The lipid identification is done by software called Lipotype Explorer, which is based on an academic software developed in the Shevchenko lab. And the statistical analysis is, and also data visualization is done by a browser-based software tool called Lipotype Zoom, but we have also many other means and tools to do statistical analysis. So as we are today discussing large-scale projects in the context of cohort studies that deal with population health, the data quality is obviously key. And data quality is essentially based on highest analytical performance. So we have to make sure when analyzing thousands of samples that, for example, the equipment is working really optimally. Therefore, in addition to a whole panel of system suitability tests that we run prior to the actual sample analysis, we also assess method performance based on reference and blank samples, which are usually run along the actual study samples. So as I said, I just want to briefly introduce to you how we check quality of large-scale studies. And as I said, we work here at Lipotype in 96-well formats. And here you see a typical plate, 96-well plate full of samples, how it could look like. So usually we distribute evenly on the plate reference samples, here shown with the green R. So we have them all over the place. And also blank samples here indicated by a red B across the entire 96-well plate. And this helps us to make sure that there is, first of all, no cross-contamination of samples. And it all allows us also to monitor parameters such as analytical drift and potential batch effects. So this is a kind of plot that helps us to check for analytical drift. So when we analyze a full 96-well plate, it takes about 8 to 10 hours, depending on the sample type, to really analyze the complete plate. And since the samples are analyzed sequentially, the last sample is basically analyzed 8 to 10 hours after the first sample. So we have to make sure that the lipid values measured are correct even after this long time. And this we can achieve by checking the lipid amounts here on the y-axis, depending on their position on the 96-well plate, which is depicted here on the x-axis. So, and ideally, the lipid amount remains the same no matter when the sample was analyzed, as it is shown here. So usually we get a nice straight line here showing us that values don't drift over the time the plate is analyzed. And the same holds for samples being present on different well plates and measured, for example, on different days. So the reference samples really help us to monitor batch effects. And again, we expect the lipid values to be similar, independent from the plate, from the mass spec, from the analysis day, or from the person who did the analysis. And this is shown here for example, triglycerides. So these are only a few of the measures that we take at Lipotype to ensure optimal analytical performance and highest data quality. And in this way, we really achieve excellent reproducibility. Technical variation is usually less than 10%. We achieve a wide dynamic range, greater than 4 orders of magnitude. Sensitivity is in the nanomolar range, and we require really only low sample amounts. For example, 1 microliter of blood plasma is sufficient to get a full panel of lipids analyzed from a blood sample. And the method is designed for high throughput, so we can analyze 400 samples per day with the current setup here at Lipotype. Okay, so much for the introduction. For the rest of the seminar, I will talk about a number of examples of cohort studies dealing with population health in which Lipotype shotgun lipidomics was successfully applied. And these case studies deal with, for example, metabolic disease, anorexia, diabetes type 2, neurobiology, skin health, but also cardiovascular diseases. And I would like to start actually with metabolic diseases, which are often manifested as obesity. So just to pick you up a bit, obesity is usually measured, you all know that, as a body mass index, BMI, which puts body weight into relation, in relation to body height, actually. And people with a BMI greater than 25 to 30 are considered overweight. Obese people have a BMI between 30 and 35. Severely obese people between 35 and 40, and everything above 40 is considered morbidly obese. And obesity is clearly a global problem, and that's why one really speaks of a obesity pandemic. In the United States, for example, about 40% of the overall population is obese. This is not necessarily the case in every country in the world. For example, in Japan, it's only 3.6%. So it is also a problem specific to certain societies. The whole problem started in the 80s, where we can observe really a steep increase in the percentage of obese people. So most importantly are the green lines here, which indicates obese men with a straight line, the full line, and the dashed line obese women. And this steep increase in the 80s is most likely caused by more and more sedentary lifestyle and the consumption of highly processed foods and snacks. And the problem now is not so much that people put a little more weight on the scale or don't fit into their wedding suit anymore. It rather comes with really serious health problems. Obesity can give rise to, for example, myocardial infarction, fatty liver, diabetes type 2, but also cancer, which obviously reduces life expectancy and also is a huge economic problem. For example, in the United States, again, $200 billion of extra costs are generated for the health system because of this obesity pandemic. And this pandemic actually also causes $150 billion loss of productivity due to illness, basically. So it is really a serious issue. And that's why we set out to investigate whether the plasma lipidome is actually reflecting metabolic changes that are caused by overweight and obesity. We worked together with the universities of Lund in Sweden and Helsinki and used samples from the FINRISK cohort and the Malmö diet and cancer study. We analyzed about 1,250 blood plasma samples and used machine learning to build a mathematical model for the prediction of different obesity measures. We used the data from the FINRISK study to build the model by repeated cross-validation as like sketched here, and validated the resulting model against the Swedish Malmö cohort. The obesity measures we used in the study were body fat percentage, waist to hip ratio, waist circumference, and body mass index. It turned out that body fat percentage was predicted best based on the lipidome. The model that had the lowest error during the cross-validation, here shown on the y-axis. So body fat percentage here in blue, as you see, has the lowest error. It's followed by waist-hip ratio, waist circumference, and body mass index. As I said, the model was built based on samples from Malmö and validated in the Finnish data set. And as you can see here, the prediction for body fat percentage works really well in the validation cohort, here in green, with the error being actually the same as for the training data set, which was based on the FINRISK cohort. So this is a really robust model that we could build based on the plasma lipidome. So the model is working in an independent cohort, and this validation cohort was really sampled and analyzed entirely independent from the initial training cohort, which is really an essential requirement for any biomarker identification study, and relies on a robust and highly reproducible analytical platform eventually. The model for obesity prediction is based on the analysis of several hundred individual lipid molecules. And those lipids belong to many different lipid classes throughout the entire lipidome, showing that it makes sense to really use a global lipidomics approach for biomarker identification studies. So as you can see here, all those lipids that get this asterisk here, all those ended up in the final model. And as you can see, these asterisks are really distributed across the entire lipidome. So really each part of the lipidome contributes to information about the metabolic state of an obese subject. So as many of you might know, the lipidome is hierarchical in nature. Each lipid molecule belongs to a lipid class. Let's say, for example, let's take sphingomyelin. So we have here a symbol like a single sphingomyelin molecule. It belongs to the class, of course, of sphingomyelin, and sphingomyelin in turn belongs to the lipid category of sphingolipids. So now one can use these different levels of information that we find in the lipidome to find out whether it provides additional information to the classical clinical lipid parameters such as HDL and cholesterol. And this is what we've done here in this figure. So let me take you through this. So the y-axis of this plot shows us how well obesity is reflected by different levels of information in terms of R square of a mathematical model. So R square basically stands for the variability explained by the mathematical model. The dark blue line that we see here shows the performance of the classical clinical parameters such as LDL, HDL, and cholesterol. When we now add lipidomic information to the model, the prediction of obesity gets better and better the more lipidomic details we add to the model. See here, adding information on lipid categories in light blue improves the model already a bit. It further improves when we add the information of lipid classes. This is the green line here. And it achieves really the best performance when we add lipid species, lipid subspecies information, meaning the information on the individual lipid molecules. So clearly, molecular detail is required to achieve optimal performance of this model, meaning providing really a proper metabolic description of the obesity state on the level of the lipidome. So therefore, clearly, the plasma lipidome correlates with obesity and reflects actually body fat percentage much better than BMI, for example. The lipid species detail, as I've shown just now, is really needed for accurate modeling. And this is really interesting for stratifying obesity to predict future health risks, basically. So this is ongoing work, which will be based on this study just shown. So the take-home message for the obesity part is that lipidomics, the lipidome, really outperforms classical clinical readouts for describing the lipid and obesity state of a person. It's not only better than the usual field methods such as BMI or physical measurements such as body fat percentage, but it's also much better than the classical clinical chemistry, highlighting really the potential of lipidomics to aid in obesity risk assessment and monitoring prevention programs. So the next topic we are going to discuss is quite the opposite of obesity. It is anorexia nervosa. Anorexia, as you might know, is an eating disorder that is characterized by low body weight, food restriction and body image disturbance, fear of gaining weight, and the desire to be thin. And it typically affects teenage women or young adults. Treatment involves the restoration of a healthy body weight by refeeding programs and the treating of the underlying psychological problems as well. But the priority really has the restoration of weight. And in a collaboration with the University Clinic here in Dresden, we investigated the effects of refeeding on the lipid metabolism. So this study included 39 female patients with an average age of 16 years and an average BMI of 15. 23 of the patients finished the program for short-term weight restoration and reached a BMI of close to 20, which is a 26% increase. And in a control group, participants were of the same age and had an average BMI of 20.5. The refeeding involved a daily caloric intake of about 2,000 kilocalories, consisting of 3 meals, 3 snacks, and a fluid intake of 2 liters per day. The refeeding program took about 85 days on average, and plasma samples were collected and lipidomes were analyzed. And we then performed statistical analysis in order to compare lipid profiles between anorectic patients before and after treatment, and with healthy control subjects. And we also compared the lipid profiles of patients before and after the treatment itself. And the analysis revealed, I'm not going into details here, but the analysis revealed an increase in ceramide concentration after refeeding, higher cholesterol concentrations, shorter and less saturated triglycerides, And most interestingly, when we applied our recently established mathematical model for the prediction of body mass index, the one I've just talked about, on the lipidomes of the anorexia patients, the predicted BMI was much higher than their actual BMI. And this strongly suggests that that there are adverse effects actually of the current refeeding practices in the lipid metabolism of anorexia patients. And the clinicians whom we work together with, they suggest, or they say that these results really suggest that one should rethink these refeeding practices. And but also what's clear is that more follow-up studies are required here in order to investigate the long-term effects of the refeeding to really substantiate these conclusions. Okay, after two examples dealing with basically eating disorders, my next example deals with a neurological disease. And is about a twin study that aimed at the identification of lipid biomarkers for multiple sclerosis, which is done in a collaboration with the German Center of Neurodegenerative Diseases. Multiple sclerosis is a chronic inflammatory and neurodegenerative disease of the central nervous system. The body attacks actually the myelin sheath of the neurons, as you can see here, in the brain and the spinal cord. And the damaged myelin causes communication problems between the central nervous system and the rest of the body. And there is no cure for this disease today. This is the brain MRI scan of a 30-year-old man with multiple sclerosis. The white signals here, for example, and here are multiple lesions in which myelin structures are destroyed. And since myelin is rich in lipids and lipid metabolism might play a role, that was the hypothesis, in the disease, but also in the process of remyelination. And that's why we were wondering whether changes in lipid metabolism can be detected in blood samples of multiple sclerosis patients. In this cohort, we analyzed plasma samples of 73 twin pairs who were discordant for the prevalence of multiple sclerosis. And the beauty of the study really is that we can control for genetic effects by conducting a paired statistical analysis of these data. And what we found is a significant reduction of ether phospholipids in the multiple sclerosis patients, both on the lipid class level, as you can see here, PCOs and PEOs are ether lipid classes, but also on the individual lipid species level, as shown here in this volcano plot. So just to briefly tell you what these ether lipids are, they are characterized by a fatty alcohol, shown down here, which is attached to a glycerophospholipid via an ether bond, in contrast to the usual ester bond we have in, let's say, normal phospholipids. And these ether lipids are usually very rich in polyunsaturated fatty acids with many, many double bonds, as you can see here. And these changes we observed, as I said, were most prominent in ether phosphatidylethanolammines and ether phosphatidylcholines. And the fatty acids that were attached there were usually arachidonic acid and docosahexaenoic acid, polyunsaturated fatty acids. And these function as precursors for the synthesis of eicosanoids, which are lipidic signaling molecules that play important roles as mediators of inflammation. And we therefore interpret the observed reduction in ethers as a general consequence of prolonged inflammation, which basically consumes the polyunsaturated fatty acids that are attached to ether lipids. Although clearly multiple sclerosis specific pathophysiological role here cannot be excluded based on the data, but nevertheless it is an interesting observation. Okay, so far we have discussed 3 cases dealing with cohort studies involving blood samples, but since blood is just one sample type that can provide information about health and disease, I would like to play now a short intermezzo on skin health. So the skin, as you all know, is our outermost, especially its outermost layer, is our barrier to the outside world. And the sebum and the stratum corneum are very rich in lipids, mainly triglycerides in the sebum, which is the greasy film on our skin, and cholesterol and ceramides in the stratum corneum itself. And the skin lipid profile is affected by many different factors, such as age and sex, but also by the season of the year and skin diseases. And the skin lipid profile in turn influences functional skin parameters like hydration of the skin, pH, morphology, and barrier function. And we have developed here at Lipotype a technology for the molecular analysis of skin lipid composition. And it is based on a very simple sampling technology, so-called tape stripping, as it is like depicted here. So we simply put tapes on this person's skin and peel it off. The lipids of the sebum and the stratum corneum stick to the tape and can be done on any body part. And we then simply extract the lipids from the tape for mass spec analysis. And by means of this technology, we conducted a baseline study on skin lipid composition of more than 100 subjects. And among many other things, we found that the lipid content of the skin is changing with age in females. So this is total lipid content plotted against age. And you can see here the solid line, a drop in lipid content for females with age. And while for males, this value remains rather constant at a lower level. But we don't only see a change in overall lipid content, but we also see compositional changes. And again, especially in females. There is a strong reduction of triglycerides here in what is this, orange, yellow. And while at the same time, the cholesterol amounts here in blue increase. The ceramides in green, another important lipid class in the skin, stay constant. For males, we see similar trends, but it's not as pronounced as for the females. So in summary, we observed age and sex specific skin lipid compositions. We see body side specific lipid compositions and also skin layer specific compositions of the skin. So these I haven't shown, but these results really have important implications for the classification of skin types and also for study design in dermatological cohort studies. And these sort of studies are currently ongoing with different focuses, skin diseases, different skin types, and so on. So this is work that is about to be published hopefully soon. Okay, let's now move back to metabolism. I know we have a lot on the plate today, but please bear with me. It's worth it, I think. As a reward, I will show you the sweet chubby cat that I've shown you earlier later again. Let's talk about diabetes type 2 more specifically. And in our most recent work, which is under revision currently, we've established a lipidomic risk score for the estimation of the risk to get diabetes in the future. And I would like to tell you a little more about how we've done this. So in the Malmö Diet and Cancer study, which I've mentioned earlier, blood samples were collected in the early 90s, as you see here. And the volunteers were followed up for the next 23 years until 2015. And they were followed up for diseases like cancer, cardiovascular disease, and of course also diabetes type 2. In total, we have analyzed more than 4,000 subjects, and about 500 of them developed diabetes actually. And we measured their lipidomes at baseline and used the data to identify a lipid signature for type 2 diabetes and build a risk score for predicting the risk of developing diabetes in the future. And we compare the performance of this risk score against a polygenic risk score. So what is a polygenic risk score? It is based on so-called SNPs, single nucleotide polymorphisms, which are essentially point mutations in our DNA. And it's known that certain SNPs are associated with a risk for getting a specific disease, but in fact, it's usually the combination of many, many different of these point mutations that are used to calculate the risk of getting a particular disease. And this is so because most diseases are polygenic in nature. And that is why this risk score, because many SNPs are involved, is called a polygenic risk score. So with lipids, we can do a similar thing. We determine that which lipids actually are associated with developing diabetes in the future. This is shown here. And we use these to calculate a lipidomic risk score in the same way as it's done for polygenic risk scores for each individual in the study. And we then, and this is shown here, we sort these subjects according to risk score and calculate the diabetes case rate in each of the deciles of this whole population. And as you can see here, in the highest decile, those with the highest risk score, we have a case rate of about 25% based on the case rate for diabetes, for getting diabetes in the next 23 years with a polygenic risk score. While for the lipidomic risk score, we have a case rate of something close to 40%. So clearly, lipidomics predicts the risk for getting diabetes much better. So that means we have in this study identified a prognostic lipidomic signature for type 2 diabetes and could calculate a lipidomics risk score that outperforms the polygenic risk score. And that means it predicts the diabetes risk much better. The reason for that is most likely that the genotype just tells us something about our predisposition for getting a disease, while the lipidome, which is actually a real phenotype, shows us what's actually going on in our bodies and in our metabolism. Okay, that leaves us with cardiovascular disease. And in this last example, I'm going to highlight the power of combining different omics technologies, in this case, genomics and lipidomics. So cardiovascular diseases like stroke and heart attack are nowadays the leading causes of death. So we teamed up with researchers from the University of Helsinki in order to study the interplay between genes and lipid metabolism and their relation to several cardiovascular diseases. The work resulted in a paper on the genetic architecture of human plasma lipidome and its link to cardiovascular diseases. And again, as in the risk score, the genomic differences between the study participants was assessed based on SNPs again, so based on a GWAS study. And the combination of genomics, so the GWAS study, the genome-wide association study, and lipidomics allowed us to calculate to what extent lipid levels are genetically determined. And I'm showing only one part of the results, and this is one major outcome of this analysis, that the levels of polyunsaturated fatty acids, like for example arachidonic acid, depend significantly more on the genotype than the rest of the fatty acids. So here we have 3 kinds of polyunsaturated fatty acids, and you can see that in comparison to all the other fatty acids, we get a really significant difference in the heritability estimates that we can calculate based on the genomic information and the lipidomic information. So there is clearly a genomic component to the lipid amounts that we measure in our blood samples. We also found that the SNPs actually have phenotypic consequences regarding changes in lipid levels. And shown here is an example for one sphingomyelin species. This particular SNP is associated with the incidence of cerebrovascular diseases, shown here, which is characterized by damaged blood vessels in the brain, resulting in a so-called mini stroke. And this really shows that the combination of genomics and lipidomics is quite powerful. It has the potential to identify causal relationships between genetic markers and a disease state by determining actual physiological changes. And in this case, in lipid metabolism that are associated with a specific genotype. So we could identify in the study many more of these associations, which really have the potential to deliver information for novel drug targets and disease prevention strategies, because of course all these SNPs are associated with a locus on the genome, meaning a particular gene, right? And so this is really interesting data for disease prevention strategies, such as, for example, lipid metabolism altering therapies. Okay, so that was my last example for the use of lipidomics in population health studies. And as promised, here's the cat again. And before I finish, I would like to make a few last concluding remarks. So why do we think that population health studies must run lipidomics? Back to our initial question. First of all, our shotgun lipidomics technology provides really quantitative results with excellent technical reproducibility for hundreds of samples. So it's a very reliable technology. It requires only very low sample amounts, and it is a high throughput technology and can deliver results within weeks. So one really gets data right away and can go with statistical analysis and data interpretation. An important fact is also that the lipidome is stable in samples that are stored for years if they are stored properly. That also means there's a real value in legacy samples that are sitting in freezers for example, in retrospective studies. Various study designs, like for example, as I've shown, twin studies, but also intervention studies with crossover design, cross-sectional studies, longitudinal studies are possible and can be done and have been done also using lipidomics analyses. And finally, lipidomics really enables disease stratification and risk assessment based on molecular information that goes way beyond the classical clinical chemistry. And I hope I have shown you that there is really information, added value in the lipidomes that have the potential to also change disease diagnosis, prognosis, and also risk management. And with that, I'm done, and I would like to thank you for your attention.